Hello everyone, I'm Dr. Amit. So in our last video we discussed what collagen is and its various type. Now we are going to dive into the fascinating process of how our body actually builds collagen. Okay, so we'll talk about collagen synthesis and its structure and the important clinical disease that occurs when any of this process goes wrong. Okay, so the total steps are seven. Okay, synthesis, hydroxylation, glycosylation, exocytosis, proteolytic processing, assembly and alignment, and cross linking. So we'll learn all these steps one by one. Okay, so the first step is synthesis. Okay, so in this step, translation of collagen to form a pre-pro collagen. Okay, so this involves a specific sequence of amino acids. Okay, the amino acids are glycine, proline or lysine, plus hydroxyproline or hydroxylysine. Okay, and at last the structure form is pro alpha chain. Okay, so this is the first step of collagen synthesis. Now we'll talk about the second step, that is hydroxylation. Okay, so in this step actually the hydroxylation of proline or lysine occur. Okay, and this step is vital because it requires vitamin C. Okay, and if there is deficiency of vitamin C, then scurvy will occur. So most of the time the MCQ asked is if there will be defect in hydroxylation, or if there will be deficiency of vitamin C, then which disease will occur, then that scurvy will occur. Okay, so we can see the structure of collagen here, so that 2-OH group is added in this step. Now we'll look in the third step, that is glycosylation. Okay, so in this step glycosylation of pro alpha chain hydroxylysine occur. Okay, so we remember that in this synthesis step pro alpha chain, okay, this pro alpha chain formed, and this in this step in the third step of glycosylation, this glycosylation of pro alpha chain hydroxylysine occurs. Okay, so we'll now talk in details. Okay, so in this step pro alpha chain hydroxylysine, okay, and it form pro collagen. So this is pro collagen now. Okay, and there will be formation of two bond, one is hydrogen bond and one is disulfide bond. Okay, and the final form appear will be triple helix. Okay, so this triple helix will form. So the most important point, or from the MCQ point of view, we must remember that if there is problem, okay, if problem occur in this step, glycosylation step, so there will so triple helix will not form, so that will lead to a disease called osteogenesis imperfecta. So when we will talk about osteogenesis imperfecta in detail, we also talk about this step. So if there will be problem in triple helix formations, or if there will be problem in glycosylation, then it will lead to osteogenesis imperfecta. Now we'll talk about the fourth step, that is exocytosis. Okay, so now glycosylation occurred. Okay, now once the internal processing is done, okay, this pro collagen formed. Now the pro collagen comes into extracellular space. Okay, and this is so this step is called as exocytosis. Okay, now the fifth step is proteolytic processing. So whenever we see this word lytic, okay, this lytic, so this lytic means cleavage. Okay, so there will be cleavage of the disulfide rich region. Okay, so we all know here we read that two bonds, hydrogen and disulfide bond form. Okay, so in this proteolytic processing this disulfide rich region get cleavage. Okay, and it form insoluble tropocollagen. Okay, now we'll talk about the sixth step, that is assembly and alignment. Okay, so in this step whatever the collagen has been formed, okay, those collagen will assembles. Okay, those collagen will assemble, and so collagen assembles and fibril aligns by the cross-linking process. Okay, and the last step is cross-linking. Okay, so this step, this cross-linking step actually is the final step that develop a strengthening of these fibrils. Okay, so these fibrils will get more strength okay. And in this step, enzyme uses lysyl oxidase, and this enzyme use copper, okay. So sometimes MCQ also ask which enzyme is used, lysyl oxidase, and which element, okay, is used, that is copper, okay. So the most important point which we must remember from this step is, if there is problem in cross linking, or if there is problem in this step, then a disease called Menkes disease occur, okay. So if there is a problem with cross linking, it will lead to Menkes disease. So this is all about collagen synthesis, okay. That cover the collagen synthesis and the clinical condition associated with each steps, okay. So if you like this video, please subscribe to my YouTube channel and stay tuned for more updates. In the next video, we'll talk in details about osteogenesis imperfecta, Ehlers-Danlos syndrome, and Menkes disease. Thank you.